So thanks, Kendra, for your kind invitation. It's great to be here and tell you about this work. As you said, yes, I've been conducting kind of a range of activities related to setting up some of the basics in the model, using primarily zebra finches, but also doing quite a bit of comparative work. Focused mostly on understanding vocal control and vocal learning pathways and mechanisms. And so I'm going to start with the tape that was recorded in the lab, which basically describes, is it work? I thought it was. Okay. It's kind of a little snapshot of the life of a zebra finch. This is in the lab, but
a juvenile zebra finch is about to be exposed to the song of an adult male. A female is introduced into the chamber, and in response, shortly thereafter, the male begins to sing. The juvenile, 4 to 5 weeks old, is paying close attention. Like a human baby learning to speak, the juvenile will try to recreate the sounds until he gets them right.
So it's nice because the kind of description, well, you see in the male, the female, the juvenile, and they kind of pushing the case that the analogies with the human speech acquisition, which primarily based on this several analogies in the process. And there was a very brief attempt of the male to mount the female. And so many interesting questions arise, primarily the question of how the juveniles acquire the model and then are able to modify their song by practice of the song. And basically, this slide is taken from work of Scharf from early. This shows a sonogram or a spectrogram of the song with the various the various syllables, the spectral components here, and the temporal components. They create the syntax. And this is the song of a tutor. And juvenile birds, in this case, around day 30, you can see how they start with a very sort of immature song, and then different elements will gradually be shaped and turn into something that resembles a lot the adult song. And this is a very active process that requires intact audition, presumably an auditory phase formation, auditory memory phase where the song of the adult is memorized by the juveniles, and then the juveniles try on their own devices to replicate that sound using production circuits through auditory feedback. This is the core of the vocal learning process, and we believe it's a very rare trait in evolution, and I'm going to be showing slides like this a few times. What is shown here is that songbird possesses this trait, and this is an old phylogeny. I'll go back to that, but other groups of birds possess the trait as well. We have here hummingbirds and parrots, and initially it was thought that these three groups evolved this trait independently, but now there's been revisions of the phylogeny, which I'll get to in a moment. And among mammals, we know, of course, vocal learning provides important basis for speech acquisition in the sense of acquisition of novel acoustic elements that can be used for the development of language. It's pretty well established, at least in terms of dialects and new vocalization acquisitions in whales and dolphins. And there's also some reasonable evidence, especially in terms of dialects in bat species. So it's fairly sparse, though. There are more anecdotal evidence in a few more groups, but I believe this is the strongest evidence in mammals. I'll go back to that as well. But overall, it's a rare trait in mammals, fairly prevalent in birds, especially if you consider that songbirds, by some accounts, are responsible for perhaps half of all the avian species in the world. So even though the majority of orders also do not express the trait. So again, is rare, and as far as we know, rodents and non-human primates do not have vocal learning, and now we can get back to that and discuss in more detail the extent to which some reports suggesting some sort of a primitive or primordial form of vocal learning, or at least adaptability in these other groups. But by far, there's no convincing evidence that rodents or non-human primates are capable of imitative learning in a similar extent to what songbirds can do. Several of the properties that bird song learning expressed, and they're similar to speech acquisition. Again, the need of auditory model to shape their vocalizations. A critical period for sensory acquisition, especially in birds like species like zebra finches. Need for auditory feedback to guide the practice. The babbling, something analogous to babbling, those immature vocalizations early on that then get shaped into more complex patterns. Individual variabilities tend to be a signature as well of vocal learners, dialects and so forth, and the learning of some sort of temporal structure that some might argue it's more like a phonology type of thing versus a more complex syntactic organization. But anyway, these are, they're all things that are present in songbirds and that resemble the human. And I'm going to talk today a little bit, expand a little more similarities in the circuitry and the molecular genetics of vocal learning and related pathways. To get started in discussion of the brain and pathways, I want to remind those of you who know about this and tell you about those, to those that are not familiar with the big nomenclature revision that Ken referred to. This came from the realization that early assumptions about the avian anatomy were wrong. They were based on the assumptions that because of a simpler architecture organization, apparently simple architecture organization of the telencephalon, the bulk of the telencephalon depicted here is in this purple color was homologized into basal ganglia structures in mammals. We now know that is not correct based on a combination of molecular connectivity data and so forth. And only small regions were thought to be kind of homologous to corticoid, including the hippocampus here. We now know that about 85% of the telencephalon approximately are probably homologous as a field to areas that will give rise to cortex, amygdala, and claustrum, such structures in mammals. Birds do have a basal ganglia. That basal ganglia account for about 10 to 15% of the volume of the avian brain. So this is a more, what we think, a more accurate depiction. And what's really interesting is that then you're left with questions about the organization of the pallium here. Again, it doesn't resemble the cortical layer structures for the most part that you see in mammals. But these cells have the same probably origin, again, as a field of these areas that give rise to cortical and basal and amygdala structure. So that led to this big revision in terminology. All these areas were used to be called striatum in the past, and now they're called pallium. And a lot of the important literature in the field still had used this literature. And if you go to early years, mammalians would think that all avian brain researchers will look at basal ganglia. That's quite unfortunate because some of this has been clarified. But again, some of the classical papers in the field, that nomenclature is still there. It can be still confusing. So it's important to take these things into account. Now, so taking these things into account, the majority of the telencephalon is paleo. It's like cortex-like with a different cytoarchitectonics. Now we look at the organization of the vocal control pathways. This is a simplified diagram and showing that the areas that control singing behavior and vocal learning in songbirds can be divided into these two pathways. We usually refer to these more caudal pathway as a direct vocal motor pathway, and this other pathway is the anterior forebrain pathway. The direct vocal, by the way, here's the telencephalon. These three nuclei here, the HVC, RA, and LMAN, are located in that paleo area. So these are like cortical-like nuclei versus area X, which is located in the striatum. So we have both cortical and basal ganglia components involved in vocal control pathways. The direct motor pathway through projections from HVC to RA. RA has projections then to the midbrain and to the N12, which is the motor neurons that innervate the syrinx. That's the vocal organ. And as well, there's projections to respiratory control elements. And phonation, vocalizations depend on the interaction of your vocal motor organ with respiratory muscles. And all these is thought to be under the control of these direct vocal motor pathway. Lesions to any of these nuclei will abolish singing behavior in adult males. And of course, then we have the anterior pathway that also involves some paleo structures, basal ganglia, thalamic loops, And this is very important for learning. Lesions to this pathway abolish learning in juveniles and also involve, to some extent, in variability of vocal patterns, even in adults, that persist in adults. So because these areas are so discrete and the connectivity is so well known, that allows us to look at the gene expression in these areas and look at the genes involved and how they are regulated, how they are modified by various factors. And that's kind of a big advantage in a way over some other organisms, including mammals, where it's harder to have this segregation. They go back to that a little later in the talk. These forebrain nuclei, by the way, I should say that the midbrain and this brainstem areas, this is conserved across vertebrates. If you look even at vocalizing fish, you probably have analogous areas there. But these telencephalic areas, the cortical-like and basal ganglia areas, forebrain nuclei, are only present in vocal learning groups. In fact, those three groups that I alluded to, the songbirds, the parrots, and hummingbirds. All these other avian groups that are non-vocal learners, to some extent, the ones that have been studied, like pigeon, chicken, quail, owl, raptors, and I also include here subocines, which are passerines but not ocines, they're not vocal learners. These groups do not have those nuclei, so these nuclei evolved in concert with the vocal learning ability. Although these birds all have, at least we think to some extent, these brainstem, midbrain vocal areas. In fact, you can elicit vocalizations, calls like vocalizations by just stimulating here the midbrain area in a chicken and pigeon, for example. So the ability to learn depends critically on the development of these cortical and basal ganglia structures, and also pay attention to this RA to N12TS projection. We're going to get back to that. That projection is the main output. All the circuitry in the forebrain, all the processing occurs here. The final output is through descending array projections. This is a critical projection that we believe is analogous to the human projection from the laryngeal motor cortex down to the nucleus ambiguus. We'll get back to that comparison. We are very interested in the development and the properties of this projection system. So work we did in collaboration with Eric Jarvis, now beginning to be a long time ago. we have looked at the structures in songbirds using sort of typically the zebra finch. And again, this, to remind you, is I'll get back to that a little later. It's an outdated phylogeny, but just to clarify here, we have these three groups, the parrots, the hummingbirds, and the songbirds. We have examined brains of all these three groups, and we can define vocal control areas of both the direct vocal motor and the indirect learning pathway in all these three groups. Much less is known about connectivity in hummingbirds. That is still an area of active investigation, but we see the presence of nuclei that are very similar to those in the other groups, which raise really interesting questions about convergence. We believe there's a really interesting, strong case of convergence across of both the behavior across the different groups and the brain areas involved. And these areas are, for the most part, are absent in these other groups. They are non-vocal learners. In fact, just to illustrate, early on I did a lot of mapping with inducible genes. We had cloned those EGR1, ZIF268 ortholog in zebra finches, in canaries initially actually, and called it Zenk. And this is turned out to be a very useful tool because it was highly induced by activity. Here's just, we could identify brain areas activated by hearing sound. In this case, this turned out to be this NCM area. It's part of the auditory pathway. Just to illustrate, it's very robust. And Eric later adapted this to explore the vocal motor pathways, and we can basically compare birds that are singing with quiet birds, and we can see the activation of these areas and then map them. And this is very well and easier in a way in songbirds because these pathways had been mapped previously. Actually, auditory pathways not so much, but the vocal motor pathways were well mapped. The parrots, they also had been well mapped, less so in the hummingbirds. So in fact, discovering these nuclei in hummingbirds came actually from primarily a field study that we conducted with Eric Jarvis, and basically looking at singing hummingbirds and birds that were not singing or just hearing the sounds, and then doing comparative maps using Zenk, we were able to identify all these areas. So it turned out to be a very, very useful tool to define these areas. But a big change came recently with the big effort to sequence a big push to sequence avian genomes and define phylogeny and define several other things, study phenotypic traits in birds and so forth, that culminated in more than 50 genomes being sequenced, and about around 50 genomes and so forth were used to redefine avian phylogeny, and as well as several other studies that came out. And this was published in 2014 as a series of papers addressing several aspects of avian biology. I'm going to talk about a couple of these papers, but one of the important ones, important outcomes of this. And I confess I was really surprised. I thought by this time avian phylogeny is well established, and birds, since Darwin listed some of the thoughts about evolution, and birds have been well studied for a very long time. So naively, I thought avian phylogeny is well established, with perhaps some controversies. It turns out it was pretty much of a mess. This is the phylogeny based on, there was established phylogeny before all the genomic data. And I'm not going to, don't worry about all the details. I'm not going to talk about the details, but I think the comparison of the classical traditional phylogeny based on primarily morphology and so forth with the genomic-based phylogeny leads you to realize how many examples of convergence and so forth. And I'll cite just a couple of ones that I think was really interesting. One is falconiforms. Falconiform groups, if I see this, can I separate here? Actually, this is not all the, no, sorry, it's shown here. Falconiforms were falcons and eagles, which would overall group together. And with vultures, now they are thought to be separate orders. And so this presumably is a case of, so this bird of prey is with a strong case of convergence. And there are several other examples, some of you are probably familiar with this, but there's a lot of interesting, and this is not final by any means. There are also different ways to derive it, but we believe this is more strongly derived than the older morphology. For our purposes here today, this was the more interesting point here, is that with this revision, a couple of things happened. We thought, we used to think about parrots as a separate group, and now parrots are grouped with oscines as the same order. It's called Psittacopaseriformes or Australabias. There are a couple of ways to refer to these. So a couple of interesting points. These are two of vocal learners, so you might argue, well, it's more parsimonious to think of vocal learning have evolved in a common ancestor to this group with single origin. If that's true, the suboscines that I believe I alluded to, which are thought to be non-learners and don't have those vocal control nuclei, would have lost the trait. That's kind of an interesting idea here, that consequence of that grouping together of parrots and oscines. At any rate, we now have to be a little more careful about treating these as independent events. But on the other hand, hummingbirds emerge from this as a really separate group that has no phylogenetic relationship to these. So vocal learning and associated pathways in hummingbirds and parrots and oscines probably is really independent, and we should think about this as convergent elements. The majority of intervening groups, there's no evidence of learning or vocal control nuclei, although I have to say not all of these have really been tested at the level. But there's a broad lack of dialects, which is one of the first telltale signs of learning. And it's mostly absent in this majority of groups. So now I want to tell you a little bit more about a couple of questions that have guided our research, and I'll get back to these themes. We've been searching for molecular modulators of vocal learning and related circuits. What genes are active in this circuitry and might modulate vocal learning and underlie this process? But also, we also interested in a broader question about brain correspondences between birds and mammals. I told you a little bit about how the structures are different and in interesting ways. And we are curious about the molecular correspondences. Are there converging molecular patterns? And can these be used to resolve questions of homology? And I alluded to the fact that, at least our interpretation, that broadly speaking, the pallium seems to be like cortex and amygdala and claustrum, and a couple of other structures. But as I'm telling you this, you realize one-to-one homologies are really unclear. So we don't know. There are some competing ideas, such as parts of the avian pallium being a big expansion of amygdala, or and so on and so forth. I'll go back to that point. But so these are two questions that have been guiding our efforts. And
before
yeah.
you move to this, can I ask you a naive question about
Sure.
your previous? So if I have a zebra finch and it has this learning mechanism active and everything, and trying to learn from the tutor,
Yes.
and I have a tutor from a different species that actually also vocalizes.
Yeah.
Does the zebra finch
Yeah.
mimic the other one?
Finches will not, because there's also probably predispositions about the types, predispositions both about the types of sounds that you can imitate, and there may be also restrictions and limitations about your vocal apparatus and the control. So it's an interesting question, because there's good evidence, many birds do learn from other species that do capable of mimicry. There's several birds that do, they're capable, catbirds and mockingbirds, and even parrots that copy other species. Interestingly, when we look at the circuits, we don't see differences, so we don't know yet what underlies the difference, but definitely there are species differences. That's an interesting point, and we can talk about this a little later at the end if you want to discuss this more. But I want to now talk a little bit, and I'm going to talk about our gene expression brain atlas. That's a major effort in the lab. We've been to address those couple of questions, which is what genes are active in the brain, what genes are active in the song circuit, in other areas of the brain. And and then I'll tell you about several projects and insights that we gained from looking at this expression database. Zebra consists of a public repository of gene expression data. Currently, we have data from about 700 genes by in situ hybridization, by looking at expression. Many of these came from screening of markers of song nuclei, but they derive from several other sources and databases. We have a high-resolution reference histological atlas, links to genomic resources. It's been very popular since released. It's been funded especially through a resource grant from GMS. But interestingly, until very recently, we presented this whole database emphasizing the zebra finch and the bird mollusks, and basically we had markers of, as I alluded, markers of the vocal control system, markers of major avian brain subdivisions. And we submitted some proposals to expand the database. There are some papers, and people get back to us when they say, "Well, you're developing a database that's only relevant to your birds," and interesting, of course. But what's the relevance to other organisms? And we quickly realized that we are not making justice to the debate. So, in fact, before I talk more about this, just to show some examples here, this is the high-res digital images. Using kind of a nice nano-zoomer scanners, and it was like reconstruct the whole section, and showing that we have identified several really nice markers of nuclei, like song control nuclei, the HVC, area X, RA, and so we have lots of interesting things to explore. I'll tell more about this transcriptome type of things, but we also have many markers that develop that reveal different subdivisions of the avian brain, like different layers of the pallium and so forth. So this raises questions about, well, how this compares to data from the Allen Institute, for example, where you have all cortical layers and amygdala subdivisions and so forth. So in the process of doing this and standardizing how we present this, the reference atlas, we realized that, again, besides these markers and genes of physiological relevance, many of these genes are also involved in human diseases, well characterized, and this data come from OMIM, which is the repository of human diseases. So many of these are markers for several genetic disorders. Many genes are involved in neurological behavior phenotypes from the MGI database, and many are markers of structures in mammals. So all that information was there of these genes, but we were not making that available. So we decided to modify how we present the database, and this is the new release of the database. We just released about a month ago, where we now directly present that information instead of asking people to go and dig deeper and find it. So we created kind of these portals where the database is now presented, where we directly emphasize which genes are markers of specific structures, especially the markers of the song system. But also we are exploring genes that are known to be involved in speech and language disorders. And we could talk a little about here. The number is not very large, but FOXP2 is a good example. I'll tell you a little more about this. Many genes involved in the disease and phenotypes, as I said, the MIM and MGI databases, markers of the Allen. We also classifying the genes according to function and so forth, and how they have been contributed to. Findings based on the genes are part of several studies involving songbirds and other birds and so forth. And we try to highlight how these are. So these are now, it's this new version. We hopefully it's easier to navigate and find that information, explore. We also create a way to visualize attributes within each of these portals. So I'll show you some examples. If you can go to the markers of the song system, you can directly identify, for example, markers of HVC and so forth. We listed by gene. And we have all these attributes here for each of those portals. Which one of the genes, let's say this is a marker of nucleus HVC. You can also identify here genes that are that have phenotypes and there are markers of different structures. So ways to try to integrate that information and make it more accessible and more interesting. Here's another example. We go to kind of speech and language portal, and we're just highlighting here FOXP2. And in fact, for example, there are lists of FOXP2 targets from molecular screenings, and we realize, well, we had the patterns for many of these FOXP2 targets, and we can look at the patterns, how they're distributed in the brain, and we can look at all these other, other information about FOXP2 targets, for example. As probably several of you know, FOXP2 is one of the first genes to be identified as involved in a speech and language disorder in humans, is a speech dyspraxia. And that same gene, through the work of Constance Scharf, can be, if it's downregulated in the area X of the vocal learning pathway, significantly disrupts vocal learning in zebra finches. So that's kind of an example of a shared target, a shared gene that's also involved in vocal learning in birds. That provides an interesting, very interesting vocal model organism to study the biology of this gene. I highlight here the FOXP2, how these attributes are highlighted in our database. You can look at either the phenotypes, that the human disorders, or in some other more hypothesis-driven formulations about involvement. And mouse phenotypes, you can get there's actually a lot of, in this case, a lot of phenotypes, and you have to go to the gene page to get the full list, and human disorders, markers of specific areas in the finch brain, and gene attributes as transcription factors. This is an example. Each one of these genes you can navigate and look at all the information. And yeah, Songbird studies. This is the gene info page where you can actually give links to all the important links here, the genomic links like enter a gene and unsolvable and so on and so forth. And this is not FOXP2, but this is what you get to get a direct link to the UCSC browser. We can look at both gene structure and at the probes they were using to probe the expression in zebrafinch. So all the genes now in the database are direct links to the browser. And now moving away from FOXP2, this is other neurological psychiatric disorders. This is an example. We have a very large representation of epilepsy-related genes because we were looking at several ion channels and so forth for various reasons, and all that is represented here, and maybe some behavior phenotypes. So, and also markers. So here's the example now with the anatomical. We have hippocampal markers. You can find markers that are hippocampal markers in the mouse brain, and we can go then compare the avian brain and look at the patterns. And so we hope there's interesting information there. And that in fact we realize even that correlation between mouse, let's say in the mouse phenotypes and brain localization is very poorly understood, even in mouse organisms. And we haven't found databases that try to make this close correlations. And here we are comparing those patterns with the avian brain with a Songbird model. So here's just to emphasize that to develop this was important, and this is still a work in progress. to develop a reference histological atlas where we can actually go and map these areas with some confidence using markers and so forth, and then we can relate the gene expression patterns to this reference atlas. I want to show just one example here where the high resolution image and the quality of the data. This is a calcium binding protein, but it labels highly the one of the cell nucleus, the HVC, and the area X. And I think we have very high res, so we can zoom in. This image is zoomified. You can navigate, basically like Google Map type of thing. You can move around. This is like digital microscopy if you want, and we have pretty good cellular resolution. So this is high-res digital images that are captured, I think, as a 0.2 micron resolution. And then you can even make inferences about cell types. We know these are large cell types. In fact, it turned out that we confirmed that by track trace. I think I have a slide later on showing that. So I can go here and make some inferences about the type of cells that this gene is expressed because we have high quality images like this, as opposed to no expression in these other smaller cell types with a completely different cell type. So a lot of information here. So, but now let me move on. So what I want to do now is throughout the rest of the talk, I'm going to highlight some examples of using information from the database and informing as the biology of the vocal learning and related pathways. So one of the things we did is define a better definition of the constitutive transcriptome of song nuclei. And the idea was to use microarray screenings for song nuclei markers and do something that is not usually done. When you do microarray screenings, usually people use arbitrary cutoff criteria of significance that please standardize, and then they do sort of follow-up confirmations of those criteria. And you are often left with many false positives and false negatives on those lists. So what we did, instead of doing this, we use an in situ-based cutoff criteria, I'll show you in a minute, followed then by bioinformatics analysis. So what it looks like is like this. What we did here is we plotted for each of these, so screening for HVC, RA, and RAX markers. We plotted here, it is from oligo or cDNA microarrays. We plotted here in blue and yellow the distribution of oligos that were considered differential and non-differential by the significance. And this is the significance bins. And as you see the microarray criteria, there's a lot of differentials in the early beans, and then the distribution falls. Whereas non-differentials tend to be much less, and in some cases the non-differentials actually go up. But it's very hard to use arbitrary criteria to define what is significant or not. So I'm sorry, I have to apologize. I have to explain this again, because in fact this distribution is about oligos derived associated with the in situ patterns. So I didn't explain this clearly. For example, HVC, we had a set of in situs from the zebra data set that we know were positive markers, negative markers, or non-differential. So we look at the positive and negative markers, and this is the distribution of the oligos associated with those known markers. So I didn't explain this correctly. So by looking at this blue distribution are the oligos associated with the known markers. So we started with the criteria before we do the screening, before we establish the cutoff. By doing this, so we now establish cutoff where up to this bin, there's a high rate of differentials over non-differentials. The yellow ones are the ones associated with non-differential markers. So by doing this, we end up with lists of markers that where we minimize false positives. So there are almost no false positives. The number is very, very small. And that is a much more satisfying outcome. And you also notice that criteria varies tremendously with different, this is from different experiments and different areas and so on and so forth. But for each one of these experiments, this is an example of area X. We now have a really nice set of markers of area X, 700 markers of these, where all high confidence differentials can be up and down. So we believe this is a much stronger data set. And then doing bioinformatics of these, we can have much stronger confidence in the analysis. And I'm going to put just a couple of things here. A lot of specializations associated with voltage-gated that resemble cardiac conduction type of voltage-gated channels that seem to be commonly overexpressed or differentially regulating all these nuclei. as an example. And of course, there's several other insights derived from this. I won't get into all details, but we believe this is much stronger type of evidence. And I'll show you a couple of examples here. Now, as we go into specific families, we see a lot of genes associated with brain connectivity, the fasciculation of semas, plexins, and so on and so forth. And we can look at different song nuclei and look at differential sets. And a lot of interesting insights to be gained. I'll point a couple of things only. SEMA7, which is a semaphorin, is highly regulated in many of these song systems, suggesting that some semaphorins play very strong roles in axonal guidance. But then we also find very interesting things that tend to contradict. Plexin C1 is a major receptor for SEMA7As and seems to be absent in all these nuclei. So many of the rules that guide ligand receptor binding based on mammalian data probably does not apply to songbirds.
Yeah, I have a quick question about how you collect the sample for your transcriptome.
Okay.
So do you have the animal
A simple punches.
exposed
Okay.
to a sound?
In this case,
Or it was
in this screening, it was constitutive, was not singing and not activity induced. We want to remove those variables because we know many genes are regulated by song. For this screening, you want to have a strong constitutive transcriptome, things that are not independent on activity and none of these other variables. So this is adult males that were not singing. So you might say it's very artificial, but if you don't restrict that, we have a lot of other variables. And by the way, our patterns in zebra is also like that. And if you actually go to the Allen Atlas, the Allen does not define behaviorally how these animals were collected, which is a big issue. Because many people say that some of the patterns in the Allen are not reproducible. Part of it could be because the behavior conditions are not defined. So the conditions here are very strict. You might say some are artificial, but at least they're well controlled.
Yeah.
You also looked at the females, since you're not looking at acquisition song.
Yeah, so for the females and finches, it would be hard to do in adult females because many of the song nuclei are very, very small. So it would be hard. They're follow-up experiments. I think we think of the females as follow-ups, taking some of the markers and examining. And I won't have time to go probably to the talk, but we're seeing interesting examples of really strong markers in males that are not in females. But I also know of some other examples where some markers of male nuclei are also markers of females, except the female nucleus is very small. So there, it's not really a dimorphism. It's just the size is reflected. So looking this closely will help us find some genes that we believe are truly differential, which will be, but it's trickier in the female. You have to make sure that you have the females in your in situ sections, for example. So I also want to point out a couple of other things. A lot of evidence of slit robos in the brain connectivity, and these is interesting, both because they some of these are involved also in speech and language disorders and perhaps FOXP2 regulated as well. Some of these other ones are also FOXP2, but this is a mosaic, and you see lots of complexity. So this, we believe, represents a good basic description and many hypotheses about various pathways that could be under regulation. And I'll quickly show you this other one because this is neurotransmitter receptors. At this point, all the ones for which we have probes, they're GABAergic, glutamatergic, and many of the neuromodulatory. There's a lot of information here, but a couple of things come up. Look at, for example, the GABAergic, the basic subunits that make up GABAergic receptors. There's not much evidence of regulation, but a lot of the modulatory ones that probably modulate, they're not directly involved in the function receptor, but modulate. They're very, very strong markers. A lot of downregulation among glutamate, which Eric Jarvis has already shown in some other studies, but this is sort of very comprehensive. Very strong serotonergic markers as well. So this is not complete, but there's a good overall picture of the neurotransmitter scenario here in the finches, which I think is a very strong contribution of this. Now, I want to briefly talk about some signatures of brain subdivisions that also derive from this work. By looking at these sections, we can derive quantitative estimates of these various brain regions. Now, moving away a little bit from the song system to the whole brain, and we can look at each one of these genes and look at expression in different areas. And by then, we can also derive what we call signatures. We can look at now specific areas and look at the combination of all the different genes and relative levels and derive molecular signatures of these different areas. This is exactly what the Allen Atlas did in their early report. The big difference is they had resources to 20,000 genes, and this is a much smaller scale here. And we are trying to make the case that we can build profiles and that differentiate some of these areas, and we want to build a bigger database, and eventually we want to compare that with such a database as the Allen. And go back to those questions, well, are different parts of the avian brain molecularly similar to different parts of the mammalian brain? So that's kind of pretty much a work in progress, but we can try to make the case that we can generate interesting data and signatures, unique signatures for these areas. One interesting example here is that this is a good example of how this work from Ragsdale lab at the University of Chicago that was largely based on Harvey Carton's insights into how brains of mammals and birds differ. Here's a depiction of the cortex of a mouse, or generic mouse cortex, or mammalian cortex, with input to layer 4 granule cell layers, neurons from granule cell projecting to supragranular layers. That supragranular layers generally project to the infragranular layers, and then your pyramidal cells from layers 5 and 6 sending descending projections. This is very schematic, but the point is you can find markers of different layers here. And now, even though the birds don't have this cortical layer organization, they have different masses of groups of cells that are physically separated. And one of the ideas is that these different layers have been moved physically. We can identify an area that has a lot of granular cell-like type cells, and other areas, particularly the arcopallium, that was thought to have cells that send descending projections. So they hypothesized this true, then markers of layer 4 should label these areas that are thalamorecipient zones, and that's actually where they see here, the antopallium and the field L. And areas that, markers of layers 5 pyramidal cells should label the arcopallium, and that's what they saw here in the arcopallium of both the chicken and the finch. So based on this, so that's kind of very interesting. So this is kind of the input layer and the output layer. Then there's everything in between, of which we have no clue. And even I think this is a very interesting study that illuminates some parts of it, but also illustrates some complexity. But then we took this example and explored for the arcopallium. You see these markers are in the arcopallium, but they're also labeling only specific domains in the arcopallium. So we've been exploring this by taking markers from the zebra finch that labels the whole arcopallium or segments of the arcopallium. So we are segmenting basically these molecular domains, and we want to go back then later and see if they correspond to different subdivisions of the cortex and or amygdala. Because the arcopallium has been claimed to contain these layers 5, 6 of the mammalian cortex, but also some amygdala components and some commissural components. This is pretty much a work in progress. This is not complete yet. It's part of a poster at LSU SFN. But so just that's an example of some domains. We are very excited about these findings here. This is a nucleus array, and nucleus array expresses some markers that are unique to array. And we knew this. We screened and have markers of array, and I'll tell a little more in a minute. But we also see markers that label this more broader area here in the arcopallium, suggesting that array may have been a specialization of these intermediate dorsal arcopallium, which, if you think in terms of evolution, Sebocines and other birds that don't have an array, we suspect that they may have the specializations in the arcopallium, which could be perhaps a general motor pathway. So it's kind of an interesting hypothesis, and we believe that this tells us where array most likely belongs to, this part of the intermediate arcopallium, and we predict that this is an area that where array represents a specialization of the intermediate arcopallium. So we predict that these markers, the broad markers, will also be present in Sebocines and other avian brains that don't have sonuclei, and we're in the process of testing this right now.
Any idea where that adjacent area projects to? You know, RA is projecting to the tracheosyringeal portion of the 12th.
Yeah, so if they were part of a general motor path, we would predict projections to other motor neurons. Is not been examined, except that Sarah Botcher has, and others have claimed there's a AED. They call this area an AED. We believe it's maybe the same. RA receives a projection from L-man, and the surrounds receive projections from the neopallium that surround L-man. We don't know much about representation, but you might construct this as perhaps a broader motor pathway of which RA presents just a specialization. Exactly. Yeah. Right. So we're now basically finding the molecular definitions of those areas. It's quite exciting. That's more details, but that's besides this, there are other markers that are unique to RA. So that's both. They're unique, but we don't find any markers of these other areas that are not auto markers of RA. So suggesting again a close similarity. This is also a work in progress. I apologize a little bit for the detail, but this is because I mentioned that part of the arcopallium was markers of amygdala, and we were very curious because especially this CYP, this is aromatase, for those who are not familiar, the famous aromatase, and there have been a lot of studies mapping aromatase and the alpha and beta, and the mappings are very confusing in literature, and the claim was that there's an area of the arcopallium called the tinea that expresses aromatase, and this would be equivalent to the medial amygdala in birds. And I'm mentioning this because basically, I'm back to square zero. I don't think we know where the amygdala is in birds. That's an open question. Might be surprise some of you, but still a lot of controversy. We basically still don't know. And this is one of the best candidates. It turns out that, so we are mapping here in zero sections, so it's not necessarily clear. I'm trying to find ways to best show it. And it continues on the next slide. And I want to show here, the ZBTB was found kind of almost by accident. And it's actually, ZBTB is a very good discrete marker of the so-called leucisthenia in the birds. There's a little bit of a peculiarity, and for birdsong specialists, so forgive me for a minute, for those birdsong specialists will be interested in this. We believe the ZBTB20 labels the so-called tinea, which might actually turn out to be perhaps part of the central amygdala, or at least it looks like central amygdala, whereas the CYP is distinct from that. So there are two subdivisions here. The problem with this being amygdala, though, is that these are paleo areas, and the central and medial amygdala in the mammals, at least the core nuclei, are subpaleo striatal-like structures. And so that raises some concerns about where it is really amygdala. Interestingly, here, I think, sorry, this is actually switched. This is supposed to be alpha and beta are switched. This is beta. There's alpha beta at the very tip of this ZBTB positive domain, actually distinct from the aromatase. We have now, with high-res cellular mapping, we are beginning to differentiate these domains, and we hope we have a clearer picture here. So I'll be happy to discuss more the possible implications of this in terms of homologies of the amygdala, but just want to emphasize this. And how much time do you want? 5 minutes? Okay, so I'm going to gloss over a couple of things that I had here, and there's another insights into the genomics that we found not too long ago, that some regions of the genome of birds seem to be missing sets of genes, and this was found just by aligning orthologs, synthetic groups in humans and lizards, and looking at birds, and it was obvious that some of the chunks were lost, and we characterized these as synthetic clusters that were missing in avian genomes. And without getting too much detail, that raises a lot of interesting questions about the loss of these chunks of genomic material, possibly at some point in the dinosaur lineage. That gave rise to birds, and some of the loss were thought to involve mostly non-coding genome. As you may know, genomes of birds are typically about a third of the size of a mammalian genome, so it's a lot of compression. But the loss was thought to be mostly non-coding genome. But we found a good number of synthetic clusters of protein coding genes that are missing. Of these, there's a couple examples. Markers of like vglut that's highly expressed. Vglut is a marker of cortical marker. This is an example where we first wanted to say, go to the island, grab a marker of the cortex. So this, and what is this in bird? And they would tell us which areas are like the cortex of birds. Except the problem is birds lack vglut1 because it's one of the genes in the synthetic loss. And basically we could not answer the question, but notice how vglut2, which is mostly expressed in infrapaleo and basal sort of brainstem type of areas, it's actually expressed in the paleo area of birds. And actually, so this is the vglut2. So this is the marker of excitatory neurons, glutamatergic neurons, and now we think this is the marker of excitatory neurons in finches and birds in general. In fact, these have been used as a marker of excitatory. So it's kind of an interesting question. We interpret this as the loss had been followed by sort of a, or had been not lethal because there was a compensation. It raises interesting questions about more basal species or other groups, such as lizards, which have both of them, and I think we believe they have more distributed expression of both markers. I'm going to skip this. There's a, but I want to comment. So another example I want to comment, which is also in the synthetic region, that was really surprising, really important for us, is LDLR, which is low density lipoprotein receptor. It's present in chicken, in many birds, and like the human version, and it's your bad cholesterol receptor and so forth. It's very complete in chicken, humans, and so forth. In tit was the great tit was actually the genome sequence and was truncated, and that was interesting. It's suggesting a cellularization of these, but the problem is that this genome had a lot of gaps and it was hard to conclude anything. And more recently, we look at PacBio assembly, and this is the PacBio, the latest one from an effort led by Eric Jarvis. And in these assembly, there are no gaps. And you go to the scaffolds, the LDLR in Finch is like that one with the tit, 6 exons only. So this is, we believe, the pseudogene. There's not much evidence of expression, and I want a couple of comments here. So why am I showing you this, and why is it important? I think the answer comes. I'm going to go back there. LDLR is the receptor for VSVG pseudotyped lentiviruses. So these are lentiviruses. They're broadly used. So lentiviruses, I didn't know this, but those are 2013 paper. But many people are not aware of this. So your VSVGs is one of the vectors used for gene therapy and so forth. It was thought initially not to have receptor specificity. It turns out that receptors apparently are LDLR, which are very broadly expressed. These are cholesterol receptors. They can up all over the place in many cells, except our finches, it seems to be a pseudogene. And that probably explains the difficulty in generating transgenic finches, which have been a lot of struggles to do that. This is a slide that I took from Lies and Value, where been championing this field of finchogenesis. The idea is that you inject your lentiviruses in an egg and fertilize the egg, and then you get expression, a mosaic expression. As long as you infect germ cells, then this can be passed on to progeny, and you have your transgenic line. The problem with this is that this depends on your injections infecting lots of cells. And we inject this in a finch, and you get very few cells. This is extra-embryonic tissue. We get good infection outside. Within the embryonic, there's very few cells. And if the cell is somatic, it doesn't matter. It's not a transgenic bird. They have to be your germ cells. Interestingly, some of you may be aware that your PGC cells are primordial germ cells. They circulate for a couple of days early in embryos, and they colonize the gonads. So one of the ideas about using PGCs for transgenic birds is to collect those PGCs, put it in a dish, do your manipulations, then we insert in the current, and then they will colonize the gonads. So it's kind of that's one of the ideas. But the problem, though, is that the effectivity by the lentivirus is very low. You can get that. There are 3 papers so far of transgenic transgenics in songbirds. Each one of them is probably involved several hundred to maybe a couple of thousand eggs were being injected in screening. So it's really a horrible effort and very low efficacy. And we believe now we understand a possible genetic cause is this. This seems to be a pseudogene, so it's not functional in birds. There is some infectivity because there are other related receptors that can take up the gene and infect cells, but very low efficiency. And I just put this added just because I added this because this, to me this looks like an ethogram of a circadian rhythm. This is simply not. This is just a sequence of the genome from regions that PacBio reads through, and these regions are impossible for Illumina to assemble because it looks like you took a little chunk of sequence and pasted 1,000 times in the genome. Incredibly hard to sequence, and all of these, we believe, are highly represented in these microchromosomes. As regions that are still not well assembled, not represented well in the avian genomes, and I believe perhaps in some other genomes as well. This is some of the hardest. PacBio technology is now marching through this, and just because it's single molecule sequencing, it doesn't matter. There's no assembly involved. You just get the sequence. There's some errors, but look at these things. Now, this reminds me of this cyclic thing, and yeah, I just want to say one more highlight one more thing. We're going back to the Convergent story. So we have song system in multiple species. We're looking at convergent genes that evolved and are markers in all these multiple species. And there are markers like parvalbumin that label song nuclei in all of them. And we have done screenings, microarray screenings, again, focusing now on songbirds versus hummingbirds, because those are really separate in phylogeny. So we eliminate the confounds of the parrots. And we don't get in detail, but just to emphasize that some of the ion channel modulators and excitability modulators and connectivity seems to be shared ones. So it seems like the system in both hummingbirds and songbirds evolved several genes that are shared markers. And even more so with the part of that genome issue, there was a study on convergent transcription specializations that compared humans and all these vocal learning birds. And without getting details, the profiles from the microarrays were compared with samples from the Allen database for humans. There are thousands of samples of humans from various places of the brain, and the profiles were compared in a blind fashion. And the question was asked, which areas of the human brains did these samples most resemble? It turned out that the samples from an array resemble mostly samples taken from the laryngeal motor cortex of humans. And that is very nice because it fits exactly our prediction. The array represents those nuclei. It's a nucleus that projects down to N12TS, the brainstem. And the laryngeal motor cortex confers the unique ability of humans to learn. I think I have, yeah, this is that projection that was telling you. So here's the array to N12TS. And that's the projection from the laryngeal motor cortex to the nucleus ambiguous, in this case, which innervates the larynx. So we are looking at similarities in the markers of nucleus array versus laryngeal motor cortex. I was highly surprised that there would be enough signal here, but it was very clear. We get a set of about 50 or so genes that are shared, and there's in situ confirming the data. I don't have the quality of in situ in humans is not as well. It's hard to get high quality tissue. But this led one of our collaborators to ask Christine Porforce, go back here to the phylogeny, your vocal learning birds, humans, cetaceans, and bats. Bats were thought perhaps to be vocal learners, so raised some questions about, well, do they have anything like this cortical specializations of vocal control? And we teamed up with Christine Porforce in Vancouver, Washington State University, Vancouver, Washington. And we took some of those markers, and remarkably, we are seeing localized downregulation. The same thing that was seen in those. This is an in situ, it's a section. This is completely exploratory. This is a section of a bat. This is the Seba's short-tailed bat. No one has ever characterized this cortex. And the cortex is uniformic in several markers that are expressed uniformly through the cortex. And we are seeing a downregulation in the portion of the motor cortex that is now a candidate for a vocal motor laryngeal-like cortex. And Christine and our collaborators, the Yartsev, Caltech, are now placing traces in these areas with the prediction that these areas should project to the laryngeal, to the ambiguous in the bat. And if that's the case, I should say we don't see any of these in a mouse. The expression is uniform, and the mouse is not supposed to have the projection, is not the vocal learning, and so forth. That's kind of perhaps an interesting possible model. I should probably stop here. Can I do something quick? What I want to do is an experiment with this audience. Let's move away for a minute from the lab. Okay, so imagine now you are in the forest in Brazil. And listen to this. I'm going to play this again, and oops, jumped. I'll tell you when it comes. Like a triplet. These are the sounds. A fundamental frequency. The scale is very poor here is about 10 kilohertz. There's the harmonics. There's about 80 kilohertz. Well, let's just a show of hands. Who do you think made these vocalizations? I'm talking about the triplet. It's and I can tell you what I thought, but you know, who thinks there was a cricket or an insect? I think it sounds like a cricket to me. Who think? I jumped. Who think it was a frog? I gave up my quiz. I thought at first a cricket or a frog, never a bird, or something that maybe a bat. It turns out this is the guy making these vocalizations, and there was a big surprise for a couple of reasons. There's another shot. This is the fundamental frequency expanded. The fundamental frequency is fluctuating here above 10 kilohertz. There's nothing below 10 kilohertz in these vocalizations, 10 to 14 kilohertz. No bird that's been looked at so far has that hearing range. The hearing range of birds tend to concentrate 2 to 4 kilohertz, up to 8 kilohertz, 9 kilohertz. And that's above 10 kilohertz, the threshold is so much higher for volume. That's not thought to be used. So that was kind of a big surprise, both in terms of hearing ability and production. So either these birds they're singing in a range that other birds for sure cannot hear. So either they developed a specialization they can hear, presumably they can hear, but that would be an exceptionally high frequency range. But if they cannot hear, then they're using the vocal cords for some other purpose, not for communications. Quite very intriguing. So anyway, some fun thing to do in the forest, in the tropics. I highly recommend. And if you go there, take some recording equipment. You can find interesting things. So.
There were questions about the sex differences and explain why that wouldn't necessarily work because the song system is so small in females. One struggle, though, of course, and Alan Brain Atlas has addressed this a little bit, is the developmental trajectory. That's obviously very important for songbirds. Yeah. So are there any plans to address how gene changes could be occurring within the song system or outside the song system developmentally?
I did not intend to show, but let me show it yet. There were some quick ones. I'll show you one example. That's exactly, we have not done systematically, but here's one example that, just since you asked, this can be just to show, this is an example of one of the, really excited about this. This is one of the subunits, one of the modulatory units of sodium channels. Beautiful marker in adult, and another modulatory unit is the opposite, is down. In juveniles, this is non-differential, and this is not expressed. So we have a trajectory here with something that's uniformly expressed and is downregulated between juveniles, pre-song to adult, and something that's not expressed and is upregulated. I can tell you a lot about this, very excited about this, but it's just an example. Yes, to answer your question, there will be plenty of... Now, one of the things we're struggling with in the lab is we have 800, 900 markers, well characterized in adult. How do you go about development? Do we repeat the screens and get it done, or try to get more hypothesis-driven questions now going at different classes? And this is an example where I don't need to repeat the screen. You can go now and ask questions about, yes, so we want to follow the trajectory of expression and see if these changes are a developmental program or are they guided by learning and by experience and all that stuff. Oh, definitely, or also, you know, at any given age, not all birds are at the same stage. And then you can try to correlate with right. So there's a lot of possibilities. And of course, these are also prime candidates for manipulations. You want to do knockdowns, overexpression types of things. Yeah, so that's exactly. So we have already a pretty large number of genes. Some are ion channels, others are like connectivity type of things. We would predict that manipulating those would interfere with the connections of the system, those type of things. And many of these could be different in females as well, and all open questions.
we're meeting for lunch, we can just meet down here and then we'll go over for that. And thank you all for coming and joining us.
Thank you.